Thanks everyone for being here for this panel. I'm really excited about this. We are in such a pivotal moment, and I think in this initiative and in other spaces where thinking about how ML and agents just change how data ecosystems need to be and how they need to support with ML ops and otherwise to make sure science is happening at the pace, the discovery that we need. So number one, I want to thank my co-moderator, Mike, for working with me on this panel, as well as our co-organizers, Maryann, Carol, and Owen, who also organized the panel with us. Thank you so much for your time on this. Also, thank you for Jeremy and Fenna, our brave note takers. Very quickly, this introduction to this panel should take about 5 minutes. Then we'll go into themes, conversations around these kinds of topics. They'll come up again, so there's no need to go through them per se. Fundamentally, spending about 20 minutes-ish around each one, and then opening up conversations to the full group and afterwards. So during each of these different themes, we'll have questions for panelists. And then after that's the panelist is asked, any other panelists feel free to jump in and continue that conversation until we pull another question out of the hat. So with that in mind, big thank you for our panelists. You are amazing and made this panel amazing, so thank you. And I'm going to now move to Mike to finish out our introduction.
Tim, I'll just ask you to advance the slide, I guess, for the sake of that. Yeah. So thanks. It's a very exciting topic. I think the thing to emphasize here is that substantial progress has and is being made on the notion of different pipelines from data to knowledge. In fact, the recent initiative of BICAN with the basal ganglia kind of data ecosystem is really a next step in terms of data standardization, data organization, and deployment, and cross integration. We are preparing a flagship publication that summarizes all the papers that are being submitted to CEL. And as Ed had showed this sort of figure before, I don't need to sort of belabor it, but the point is that this does form an integrating kind of and a synergistic kind of linkage between many consortium kind of activities, Brain Connects, the Brain Cell Atlas community, neuroimaging, and other disease-focused consortia. Next, please. Yeah, there's a variety of studies and packages from the single cell and comparative studies, genetic tools and disease, which basically accessed and kind of profile data in human, macaque, and marmoset. And using a variety of techniques here, and the data is all available and sort of kind of archived in the data archive, the three major kind of Brain Initiative archives. These are done through these standardized data profiling centers, these centers listed below at major sort of institutions. Next. So, a key part of this is really the standardized workflows from a sort of a standardized joint sampling plan through the advances in the selections, biobanking of specimens, to data generation, to organization data, standardized data processing. And the consortium has worked using a kind of joint milestones in which we, at every step of the way, try to kind of standardize and organize information in a way which will make it most useful. This in many ways differs from previous publication packages that have done. Central to this is work on the neuroanatomy anchored information management system, what called the NIMP, which contains sequencing and specimen kind of control portals. Next slide, if you would, Tim. This work is done through Zhang's work from at the UT Health Center in Houston, and really kind of tracks the management of from tissue selection all the way through sequencing, through the standardized sequencing. And this is something that it was really essential in dealing with kind of human data and human tissues, which we had not heretofore kind of done in previous kind of consortia activities. Next slide. Yeah. So over 18 million cells have been profiled in the basal ganglia for this package using a variety of techniques shown through here. About 95% of these are in human, although there is lots of data available for macaque and marmoset across multiple modalities, and basically shown here in a novel kind of harmonized ontology called HAMBA, that we use to kind of give kind of the structure to the striatum and its related areas. This data is all available through basically the data portal and kind of a supporting data sites integrating with the archives. Next slide. Part of this is really kind of advances in kind of using this ontology and the imaging through new coordinate kind of frameworks across species, which have been kind of harmonized and aligned. We have spatial registration techniques that allows us to kind of view and sort of access and parcelate data based on these, as well as the ability to perform studies of, say, example, functional gradients, aligning sort of spatial transcriptomic data with the data from the Human Connectome project. Final slide, if you would. And so, you know, the kind of the full pipeline here toward the end is basically a wide variety of tools and resources that have been made to sort of handle and deal with this data and explore it for users. This forms part of a kind of knowledge environment which is ready for kind of a next level of kind of, you know, sort of annotation and kind of enhancement. So I think these are just points to make that we are very much cognizant of the problems and challenges of this particular panel, and we look forward to sort of hearing new ways of kind of looking and advancing our science here. Thank you.
Thank you, Mike. And also just a note to the audience, pipeline is a word that's used in this panel, and it's an ambiguous word. When we talk about pipelines, we're talking about ingesting metadata as well as data and registration of that information, all the way through storage, common processing, QCing, QA, through to being able to work with it in knowledge base platforms. So like the bigger P pipeline in this case here, not specifically workflows, although it's included. So next up is our first panelist group. So they're going to be dancing around the topic of storage and publication. And to get us started, some of these panel groups will have intro slides, some will not. The most important thing is the conversation. So in this instance, we do have a couple slides to go through at the very beginning. I think Owen's going to guide us through those.
Yeah, thank you, Tim. Yeah, I do want to make the point that we are sort of dancing around the topic of storage, publication, and dissemination. I think what you'll discover here is that the panel members are really excited about where we are in this point of history, because we've established a data ecosystem that's unprecedented in a number of different ways. And I think people have already started to convince you that we're transforming data into the ability to make discoveries in ways that just we haven't really seen before at this point in history. A lot of people here are linking that to the success of our ecosystem. Mike flashed over some slides that just barely touch the amount of engineering work and federation that's gone on, that's happened in the last few years, that has brought a lot of success to this project. And Tim, if you'd go to the first slide, I'd just like to make a distinction here about the role of archives and knowledge bases. If you follow what NIH has done with its leadership of creating these types of services, they've been very smart, in my opinion, to make a distinction between what an archive is meant to do and what a knowledge base is meant to do. So I run an archive, and what I would say is our mandate is to really be kind of immutable. We want to be durable resources. We're optimized for essentially long-term preservation and provenance. and we want to provide essentially citations into the data. And it's meant to be intentionally neutral with respect to biological interpretation. We want to overlay biological interpretation on the data, but really, in a sense, we're almost preservationists. And then if you think about what's happening at a knowledge base, it's, in a sense, when you frame it the way that I have, it almost becomes exactly the opposite. They want to make use of our accessions. They want to make use of our explicit versioning. But the idea is that knowledge bases, in contrast, are meant to be necessarily interpretive and evolving over time. So we want them to support, a type of biological abstraction and discovery. We want to enable changing annotations and things that, our learning is going to change over time. So what they do is going to be changing over time. And then if you start thinking about that in the context of AI and model development, you also would want those to sort of adapt and change over time. That's going to be sort of a necessity. And so then the question is, well, how do these interplay? So Tim, the next slide. What I thought I would do is encourage everybody to be thinking a little bit about there's a few different planes of essentially data sharing that's going to be necessary as we move into our kind of AI readiness era. And I'll just outline these just very quickly. Plane number one, we want to start picking some standardized features. So, for the Human Genome Project, we use essentially genes as one of the main anchor points that's really shared between different systems. Here, I think it's going to be a bit different. We'll probably start assigning cell-level metadata. I shouldn't say probably, we are assigning cell-level metadata. We could be developing graphs or different types of associations that are really translate into being vectors that you could associate with genes. Another plane of data sharing is probably going to be happening at the level of models. Who trained this, what the annotations were used in order to generate a model. And then a third plane really could be thought about in terms of, as we develop models, it's okay if they're adapting over time, but we really have to start thinking about what is the objects that would sort of be used as for provenance and for validation. So I just want to introduce these ideas as important concepts to be thinking about, and I would like to encourage the panel to also be musing or dancing around those topics as they present. Thanks, Tim.
Thank you.
I just wanted to introduce briefly the challenges that were given to this first panel. There were two questions that were asked of us: how will brain cell atlas data be collected, stored, quality controlled, and disseminated by data archives? And what data standards need to be developed and implemented across data archives for optimal reuse of the data. And the figure on the right is an ecosystem analysis I did a few years ago that started to map the fact that neuroscience is finally coming to some agreement about some standards, and those standards are in green, and they help link together they help link together the very distributed data ecosystem that we have. And we have to remember the data ecosystem that we're talking about is not just the work that is going on in the BRAIN initiative, but there are many other archives. There were other large BRAIN initiatives that have also developed infrastructure. And if we truly want to be able to have community contribution across these grand challenge questions, we also need to consider how to be able to bring in people outside of BRAIN. So I thought that this would be a good jumping off point, and to now turn it over to Tim, who I think is going to moderate this panel.
Right. Thank you so much, Maryann. And then also, Mike, I won't be able to see hands, so please guide us if you see hands coming up for our panelists as they respond.
Okay, we'll do.
Thank you.
We have a hand here by
All right,
GQ.
GQ, go ahead.
Hi, very nice framing and important topics. I just want to, and actually it's just from yesterday's workshop, I came to some realization of one of the probably tangential important aspect. When Jan presented the basic building blocks and components of the entire brain initiative, there's Beacon, Brain Connect, and BBQS. It's meant to go from individual cells to circuits to behavior. And what kind of ontologies and taxonomies we have emphasized so far are focused on individual components, individual types and modalities. However, when, soon or right now, we need to link and cross the data from one component to another. It is in the bridging of those aspects that presents probably a very important challenge that probably invite us or prompt us to work on a unified or spanning modality and components kind of ontological, meta-ontological framework that links different entities together. For example, in the brain initiative, we talk about cell as a thing or types of things. But what's in the Brain Connect? What are the things or types of things are there? In the BBQS, what are the things or types of things together? How do they correlate with each other? And that's very important framing that's not been done and represents a great opportunity for us so that we can apply common data elements, identifiers, tracking, and actually that kind of metadata level development, ontological development has not been here yet, but it'll be serving a framing and architect that actually can help the knowledge-based development that the kind of requirements Owen already articulated, but the overall conceptual framework spanning the entire brain initiative components is right in front of us. And some of the challenges, I think, are because we don't have such one, and we probably could pay some attention to this.
Thanks, GQ. David.
Oops, do you guys hear me?
Yes, thank you.
Oh, great. So this is a bit of a follow-up on Zhang's comment, and it seems that most of the archiving setup is done in a way that it's particularly straightforward to put data on and retrieve data. But what I'd like to bring up is the idea that to get the theory community involved and to really focus on circuits that you'd like to have things put in form of a graph, which is what neuron is connected to what neuron, and of course annotated graph where each entry is the synapse between individual neurons, which would microscopic view, which is a groups of neurons or areas connected to groups of neurons. I mean, you could also have you have the average synapse, and you have something about that synapse. But this is really the way to engage the theory community. And I think the way it is now, there's no, it's not obviously set up for a analysis of circuits, and that's what will really form the bridge to behavior, because the same circuit analysis, of course, will lead to muscle output, and that will lead to sort of active sensing where you sense your own actions and you form these large-scale loops. So it's kind of a call to think of putting this in terms of a graph, like you might associate with neural networks.
Thank you. And I think that points a lot to like what are the different products that need to be created and maintained by consortia and between consortia that enable people to do analyses and downstream things. So it's a great point. Maryann.
Yes, so I think one of the things that this panel was asked to consider is the types of standards that were necessary in GQ. In looking at this and thinking about this last night, the sort of bottom level standards have been taken care of, and there's been a lot of investment in things like BIDS and NWB, but they don't really take care of the metadata. I think the Brain Initiative, the BICAN, has done a lot of work in this sort of unifying metadata. But if we really want to do multimodal integration, and our ecosystem is set up to separate those those modalities across different archives, then you really do need to think about this meta level on top of it that allows you to navigate across it. I do think that the knowledge layers that Owen was referencing are going to be very important, not just to distill the knowledge out of these data sets, but also through provenance, right, to be able to link across these different archives. So I think the area of metadata in particular, that was not an area that we spent a lot of time on, especially at the archive level, right? There's some basic metadata that are put in, but the sort of deeper level that lets us navigate across these and resynthesize, I do think needs attention.
Thank you, Maryann. John.
Yeah, Maryann, that's a great point. I just want to get back to the points that GQ and David raised. GQ, thanks for bringing that up. And David, we absolutely agree with you. And GQ, as you astutely noticed, Yang did mention this larger effort that we're proposing for the brain knowledge base, not just the BICAN knowledge base. And maybe right now isn't the right time to talk about it. Perhaps we can talk about it at the end of the conference tomorrow. But I think right now it's just a matter of biting off what you can almost chew now. And I think but with an eye toward this larger integration. I mean, ultimately, I think over the next 5-10 years, we do want to see an integrated knowledge base that covers not just cell types, but also connectivity, and ultimately not just circuits, but also circuits of behavior. And that was the basis of us proposing this brain knowledge base that actually spans all these things. So it's both a challenge and an opportunity. It's a huge lift. We're certainly not going to get to all the right questions to ask today, never mind the solutions. But also, I think brain is perfectly situated because we have these 3 programs ongoing. We actually have a leg up on all this. And of course, BICAN is the most mature of these 3 programs. So, I don't want to interrupt this discussion too much, but as folks are thinking about these critical issues for the BICAN knowledge base, it's also, I think, good to keep in mind that eventually we do want to link it across these other projects and modalities, and ultimately to link this to circuits of behavior. So just a little plug to, while we're thinking about these important, not small issues, there's also going to be larger issues that we hope to get to over the next 5-10 years.
Thanks. Dawen.
Yes, thanks, John. I think you brought a very important point of connecting different consortiums and how to unify these knowledge base. Currently, I think the sequencing ecosystem has been pretty standardized, and then has the hardware and software resource that can unify all the information processing pipeline. But if you look at different modalities or different consortiums, it seems like anatomy of individual neurons or at the group of a structure level, that would be kind of the bridge that can be used to bridge between different modalities, and then at least at the cell identity level to help us to bridge them, and so that we can do imitation, or we can do a kind of like a holistic analysis. But currently, obviously, because the nature of the image data, it's themselves a very large, various resolution, and so on and so forth. So I think that presented a huge technology challenge presented in front of us. And then secondly, is that from the annotation, there are just way more different processing pipelines, way more AI tools to do annotation than the sequence, one-dimensional sequence themselves, right? Because these are 3-dimensional now instead of a 1-dimension. So I just want to brought this up that emphasize on we probably need to get a more collective and focused discussion on how to integrate software and hardware and existing technology to kind of use it to visualize the raw data, annotation on top of it, but the raw data be the minimally processed, and then at the back end, resource-wise, probably have enough computational power to do on-the-fly, small-scale processing, for instance, and then the AI folks and then the computational people would still get back to that raw or minimally processed data, and then evolve AI models, validate AI models on this platform over time. So I just want to put up this point so maybe people can discuss further. Thank you.
Thank you, Dawen. Qing.
No.
Great. I just wanted to follow up on Dawen's comments and discuss a very, let's say, specific example of spatial transcriptomics as one kind of imaging modality. So I think the examples yesterday that Roel noted about how sequencing is a nice analogy. I would say the analog for spatial transcriptomics is, we also have molecular resolution, points of where these transcripts are detected. So that level of transcript resolution point cloud information is, I would say, 1 degree of resolution higher than, let's say, the cell count matrices that a lot of people tend to release. But on the flip side, I would argue that, let's say, for even FASTQ, sequencing data, at some point that was imaging data. Nobody really processes the pictures of dots of red, green, blues anymore. We just get, the ATCGs. But that being said, yeah, to Dawen's point, in theory for spatial transcriptomics, we do have that raw imaging data that could be reprocessed. And I'd be curious if, what does the community think constitutes raw data? Is it the raw, not yet decoded images, or is the transcript level molecular resolution position sufficient? And then with that, again, I think people tend to release only the count matrices, which has been actually quite frustrating for my group, because we are very, very curious in, like, let's say, the way that we would use this kind of BICAN knowledge base might be, okay, we want to know all the cells that are, let's say, connected to this region of the amygdala, and for the excitatory neurons in that region, can I pull out all the spatial transcriptomics information that has subcellular resolution, and then use that to look for particular distributions in the RNA transcripts that might be indicative of formation of stress granules or other types of biomolecular condensates or something. So yeah, I think that's kind of the way that my group imagines querying this type of data. And yeah, I guess in that sense, two points of discussion for the group is what constitutes raw data, and also the extent to which we might query the knowledge base to address various types of questions.
Thank you. Dawen, if you have a quick response, that will be helpful, and then we can move to next space.
But I do think, Tim, the panelists need to be given a chance to weigh in on these, and so allowing them to make their remarks would be good.
Yeah.
Yep. Okay. So yeah, Jin, thanks for bringing this up. Very quick, just like what level of raw data that you get. So here is a portal that we developed for rejoin, also supported by BICAN. So I just want to show you, these are actually possible with current technology. Just use neural glancer as the front end. And here you can see that spatial transcriptomics like data annotation on top of 3 different genes here, PV, GAT1, and SST in 3D.
Oh, I think we might be seeing the wrong screen.
Oh, you're seeing the wrong screen? I'm sorry. Or not really shared. How about now? Can you see that?
Yes, that's better. Great, thank you.
Okay, so yeah, so here you can see that this is using neural glancer as the front end, and we develop a back end to support hundreds of terabytes of data in one size. But basically here is very similar to what people normally see in those 3D annotations, and this is individual raw data spots and overlap on top. You can see the identification of these spots. So if anyone's interested, be very happy to discuss further.
Awesome, thank you. This is a lot of these topics are very connected, and so it's hard to sort of separate them out. But nonetheless, I'll sort of try to keep us a little bit in bounds of different themes, so we touch on everything. So I think some of the things that we heard as well are a little bit about what is data and also imaging data. And so to have a voice speak that hasn't had a moment yet, maybe Alan, we've had a lot of success stories around models and AI working with images. It's very important. We just saw that. It's very important. what are your thoughts around sort of like the challenges that we currently have with the infrastructure and like how to move forward in future space for arbitrary images across thousands of data sets that we have?
Yeah, thanks, Tim. I appreciate the question. Yeah, I think, one of the big challenges here is even with well-developed, human-based analyses, right? Image processing is a very iterative process, right? So we have big challenges with regards to doing AI analysis. Anyone who's worked with these models, even the ones that work really well, like cell pose and segment anything, they still work differently across different modalities. They will vary even within individual microscopes and imaging parameters. So I think it's really important that we have ways for these AIs to do iterative analysis. In terms of this is important also with regards to, the models. Do the models have access to the data? Do the models know how to find data across the archives? A lot of this comes down to fair AI-ready data. We talked a lot about this here recently. Are we deploying the most recent innovations with regards to AI discovery, things like model context protocols that have access to data across different archives, so that when we want to do agentic systems with LLMs for autonomous discovery of data, autonomous analysis of data, are they well positioned to be able to find, understand, and iterate over the data? I think it's really important in the early on here, right, that we have models that we can trust. We talked a little bit yesterday about the different maturity levels of these models. I think many of the image processing approaches are sort of in the infancy. They work very, very well across, again, individual modalities, individual parameters, but sometimes don't scale out as well as they should. There's also other considerations with regards to scaling out just in general. As our imaging data sets become extraordinarily large, we have to think of ways to break up those image analysis tasks across individual computers or many large scale machines and still reassemble them and keep a coherent picture of the underlying data. So I think there's a lot of really interesting challenges. I think this group is, as what we saw yesterday, what we've discussed even today, there's a lot of really fantastic innovations that have come across that are going to allow us to curate the training data, and try these models out across these diverse data sets. But I also think there's a long way to go, and it's exciting to see what we're going to develop.
Yeah, I think it's really important for us as data ecosystem to be thinking about sort of like how are we supporting models, right? Because being able to support the training of models and validation of models, having benchmarks potentially from our community easily accessible and being able to leverage, thinking about latency, thinking about high access data sets that are important to training, for instance, and the costs around them. Those are all like critical in enabling our scientists and scientific community to be able to leverage and reuse the data that we have. So those access patterns, even through the life of the MLOps, the lifespan of the model is very important. And even depending upon where the model's at, it might require different access patterns, right? And also storage requirements as far as hot or cold, whatever it might be. So to wrap up this piece and move to a next piece, which won't matter too much because it's all very connected. One last question in thinking about access patterns and like user experiences with our data archives, because I think we will need to reevaluate what are the access patterns and user experiences we'll need associated with them, right? Our repositories to really bring value in data reuse, especially in a agentic and model building world. Like, what do we think about, how do we think about the UX of data access and what could our future be for our archives? So maybe Brock.
Great, Tim, thanks. Yeah, it's an important question. I'll start by saying at a high level, the archives and knowledge bases are, should be designed to be user-friendly for the community, both for, folks contributing data and for those looking for and accessing and using the data. It's important to say the Brain Initiative did a good job thoughtfully resourcing different archives and knowledge bases for the different data communities. But even within each, there's all these different data types. Raw data was brought up, process data, annotations, metadata, various like materialized data products, for specific studies or time points or versions. And all these are useful, for different scientific workflows and tool development, AI model training, AI agentic workflows. So to your question about user experience, making these data available in multiple ways for different users and use cases and needs is critical. So programmatic access and APIs, direct data access and download, LLMs and AI assistants, thoughtful user-focused front-end design, coding examples, web-based notebooks, documentation, all that kind of stuff, all to allow users to navigate the archives and knowledge bases through different tools for searching and access and visualization. Neuroglancer was mentioned, and analysis, and including progressive workflows, so users can, get query results from that, thumbnails, subsets of data. Alan brought up subvoluming for different, enabling different AI workflows where, you have a very large data set, you have to do it kind of piecemeal. And so, I think the speakers did a good job of kind of highlighting all this stuff earlier. And to echo those on the panel and previous comments, data standards and schemas are all enabling for this machine readable metadata, again, coding examples. And I think, the archives and knowledge bases working with the phase 1 and phase 2 programs, the BRAIN programs and their consortia have, helped prioritize some of these choices that enable these workflows. We're obviously moving a lot toward these AI-based workflows, agentic workflows. And I think, again, good systems engineering for BRAIN, a lot of them like requirements-driven design toward those missions, those various programmatic goals. But, just thinking moving forward and with these new needs, and some of them are brought up by questions earlier from folks weighing in before the panel, we need these, we need inputs from all users on, what would make their experience better, new features, new tool integrations, new API function calls, new standards, potentially. And I think this is true for the existing capabilities and tool ecosystems and knowledge bases that are built around the data archives, but is going to be necessary, even more necessary for this future integration with AI, agentic workflows, cross-modality, cross-archive knowledge bases, and then this larger BRAIN knowledge base. So again, front and back end design, very key for this.
Thanks, Brock. David, go ahead.
Yeah, I wanted to emphasize one thing that is critical in connecting databases, and then, of course, critical then in trying to build circuits. And that's the location of the cells or the regions of interest. I mean, in some future world where there's a full connectome and therefore a complete graph, location in principle could disappear. But even that's not obvious. It's not clear if we could do proteomics and transcriptomics on cells that were fixed for electron microscopy. But right now, you want to go from a region that you may have recorded from. We have electrophysiology, and you have notions of where that region is connecting to, sort of mesoscopic connections. And you'd like to match it with, say, transcript data or proteomic data. And the only thing you really have is the location of the group of cells that you recorded from. So it has to be tied back to an atlas. There are multiple atlases now. Obviously, the Allen has their third installation. The FMOS group has their atlas. But tying it back to an atlas and depositing with a data set detailed information about how this was done so that other people can make a stab at estimating the reliability or the errors, I think is very important. So beyond all the APIs and all these very sophisticated schemes out there, which are lovely, there has to be a way to put a standard. This is a location plus minus so many microns in the AP direction, plus minus so many microns in the lateral direction. And this, one opinion but I mean, that's what's going to make the field go forward, knowing what the properties of cells are that go project to different regions and how to build circuits from those. Thank you.
Thank you. I think it's a nice way to end up this section, and we'll step into another one that actually is very relevant to a lot of the things that maybe you were saying, in which what are our requirements, data requirements for downstream analysis, specifically integration, ML, and AI. And so for this one, we have a couple questions that we can focus on. And not sure, Partha, if you would like to go first since you have a slide. We could talk a little bit about that. So there's a trend. Yep. Sorry.
If you want to read out your questions and then go to the slide.
Yeah. So there's a trend to automating analysis leading to auto population of scientific output, like using LLMs to curate information from the internet.
Tim, I meant the questions on your slide.
Oh, okay. Sorry, we have a question for Partha as well, but let me just go ahead and move forward. Those will come back. There you go.
Okay. Yeah. So I just, kind of coming back a little bit to what David and John and I said earlier, I want to move the focus a little bit from engineering to science, in the sense that a lot of what we are discussing are engineering issues. But if you think in terms of the science, in a sense, what we might want to do is to think of a circuit-centric neuroscience. Circuits are, of course, made of neurons. You can think of the neurons and be cell-centric, but circuits really underlie behavior. So one wants to be circuit-centric. I wanted to put up this slide to point to what I view as a kind of a data bottleneck of sorts that may be a useful conceptual thing to keep in mind as one is thinking about the data requirements for downstream analysis and integration and so on, which is that upstream you get a lot of raw data from different modalities, right? And this is just a cartoon, so there's a big diversity of stuff upstream. I'm not going to go into details. All know what that is. And then downstream, finally, people are doing different kinds of analysis. They're interested in different kinds of things and summarizing or making theoretical work or experimental. It's of different things they're interested in. In between, I think here's the concept that I'd like to put forth, is some intermediate level where things are not so diverse. Whether you're thinking of neuronal morphologies that you have extracted, pinned down to some atlas location, whether you're thinking of circuits, there is kind of an intermediate level of thinking and representation. And this is not going full-fledged theory yet. This is the kind of thing that enables theory. But I think this is away still from the diversity of the raw data. And I think that this level is really important, especially if one is going to do kind of advanced data analysis. You could build end-to-end pipeline that goes all the way from your raw image data volumes, up to what you want to do downstream. It's going to be very difficult for the community to engage in that, for a lot of different people to engage in that. So I think the role of the people who are producing data, making pipelines to, process the data and make it available through interfaces, is really agree on this kind of intermediate representation. And then that provides the interface for a larger community to engage, whether they are doing theory or they're experimentalists who want to know what's going on. And the three concepts are, of course, the cell, the neuron, and the circuit. The cell has types and omics types. The neuron has a morphology, and then you have a circuit. This is pinned down to an atlas. So I just put down this idea of kind of a data hourglass where things are a little bit simpler in the middle, not to the left and not to the right. That's why I made this diagram.
Thank you, Partha. Really a beautiful concept. If there's anyone that wants to respond to that, feel free to, or otherwise I'll move back up to the questions. I think this is an important concept for us to be thinking about in general through our discussions today. So, all right. Well, while I have you, Partha, maybe I can ask you the question I half asked you already. And so, there's a trend in automating analysis leading to auto population of scientific output, using LLMs to create information from the internet and literature and databases, ontologies. How do we ensure the results will not lead to loss of trust by injecting noise and hallucinations? Like, how do we think about quality control for trustworthiness? And who does that?
Yeah, I think, recently somebody mentioned to me, we were looking at these ontology development toolkits and looking at populating out some of these, if you want, ontologies. And somebody said, oh, just launch an agent. In the olden days, people used to read the literature and populate. Now just launch an agent and populate your ontology. And I was uncomfortable hearing that because, okay, now science is based on trust and honesty. And people have to be able to rely on products. So if I'm going to auto-populate, how am I going to know that it was reliable? So for me, the key concept is augmentation and not automation. You have to have the human in the loop. The scientist who is thinking about what are the kind of questions, setting the framework, and then doing the quality control. So you don't end up with these databases that are auto-populated by agents full of, I'm sorry to say, garbage. And then, not garbage maybe, it's just that you don't know which of those facts in there are unreliable, and that's disaster. So I think there needs to be some attention paid to the role of the human, to how you do the augmentation, how do you do this, and what's the workflow? Sure, we have to use automation. We have to use we have to read the literature, read the internet, etc. But have we thought through how the human is engaged? And not just as somebody who's sitting in the back office and generating annotations and training LLMs, but more upfront, in designing and curating and quality controlling the intermediate data products.
Yeah, and I think that's a really important distinction to have here is that this is not, so like there is leveraging LLMs, these kinds of things to be able to engage data, and then there is the ability to use AI for scientists to be able to engage the data. And like in that space, it's still informed, right? We're still iterating, right? And the scientist is still very much an expert. And so enriching the ability for the scientist to leverage the data, but making sure when they're in the middle that they can be a partner there and co-create, right? And be able to update the model and be able to give feedback on it, right? Because those specialists are a part of that model in a lot of ways. Their brains are, and they are in a lot of ways tweaking things and updating it so that the rest of the scientific community, who maybe specialize in other ways, has accurate information, right? Like, so when we create these big products in our consortia, we're responsible in a lot of ways to make that happen, so others can rely on us. It's an important dynamic. Thank you. Jean, and then Jesse, if you're around, love to hear your thoughts around QCing that space as well. Go ahead, Jean.
Yeah, it's a great discussion, and I really like David's comments. Thought they were very beautiful. But this idea of kind of what information do we need to provide to give the users the capacity to calibrate, the confidence in this information, and likewise, if I was using an agent to query this data, how do I know that let's say the data that it kind of grabs out is actually the cell types I want? And at least in the context of maybe transcriptomics, if again I'm querying for cells of a certain annotation, I would want to make sure that they do upregulate the expected marker genes. What were the markers that actually informed that annotation in the first place? Whether that's a human creating that annotation, or whether that's a data-driven and again upregulation of certain genes or proteins. But then also for, in particular, my very specific niche of spatial transcriptomics, for probe-based spatial transcriptomics, we would be very interested in making sure that the probe sequences themselves are available, because that allows us to decide, is this probe actually specific for the target gene that's intending to bind? Is there any evidence of off-targets potentially? So all of that kind of the information that would be necessary for me to have confidence in this data is going to be very, technology and very modality specific. So I'd imagine that, the community would need to be highly engaged in ensuring that, this type of data is available and accessible.
I love that. And also, I'm just thinking about atlases in general. Why not having evals for atlases and then getting a sense of measurement of model drift or underlying data drift? Like that would be, I think we do that as humans after we create an atlas, we check it for certain things, but what can we move to a space where we have evals as well? Max.
On the first question, how can data processing pipelines facilitate analysis, visualization, integration? Just quickly, I mean, the project is so central here that you can just define anything, right? So just provide a JSON file with every of, for example, for single cell RNA-seq, provide a JSON file and just make up something so everyone in the visualization portals can then adapt. But right now, the information has to be put together from various sources. I mean, this project is better than others, but you could just make up something and just dump everything that the user could possibly want to know about the data file. Processing pipeline versions, where the pipeline was run, when the pipeline was run, any metadata about the contents, put that into a JSON file and just push it through, and then the portals can pick it up. And then that could be used by other projects in the future too. Your standard, you could set the standards here, because I think that's very much missing right now.
Yeah, in a lot of ways, we're in a federated environment, data ecosystem for a lot of the initiative work, and thinking about a layer on top of that, that's an interface to be able to engage the data and the provenance around that data in a holistic way is, I think, very important and simplifying. Sarah.
So I just introduced myself quickly. I'm Sarah Teichmann from Cambridge in England. And I just want to give a perspective on this from the Human Cell Atlas Initiative, where we're doing what potentially has gone on here, which is basically harmonization of multiple data sources into reference data sets for tissues and organs. Obviously, the brain is like the most complex organ, potentially with hundreds of tissues. So it's, a special case in some sense, but even the heart, has 30 tissues. So there's a human community aspect, if you like, and that's captured to some extent, as you know, Tim, in the cell annotation platform, where you can have user comments on annotations, and then on the other hand, at a technical level, in the sense of how well do data sets integrate using technical parameters, of statistical parameters of silhouette coefficient type of thing. So those in some sense kind of provide one way of bootstrapping yourself towards a gold standard. And obviously there's a versioning and these creating references and these sort of quality control and trust will be an iterative process, I think, in all of these projects, for all human organs and tissues. What I feel we also need to kind of acknowledge, and some people have mentioned this, is that a standard way of benchmarking is by using an orthogonal data set. So for AlphaFold, the sequences were benchmarked on the three-dimensional structures. Mm-hmm. So for cell annotations, is there a way of benchmarking to some extent, at least, on spatial data sets, in the sense that the location kind of gives you the location and the relative position to other cells gives you some confidence or some information about annotation, or other ways. For instance, the complement of genes, gives you an ontological kind of annotation of the cell. For neurons, maybe it's ion channels. So I wonder whether it's also worth thinking about benchmarking in that context. So there's on the one, sorry, in a nutshell, there's the community context and the human, kind of in the trust and the iterative versioning, and on the other hand, there's benchmarking through orthogonal data. And that's sort of towards this conversation that's going on also in the chat about hallucinations and annotations and so on.
Yeah,
I hope that's useful. Yeah.
No, that's a great point. And I think fundamentally in a lot of ways, we're getting to a point. So one of the successes, I think, of at least of many brain initiatives is a product level thinking of thinking about what is the consortia creating, and certainly with the HCA, they've had a very big focus on what they're creating and coming together, and group work, group think, which has been amazing since it's global. And so, like, fundamentally around those things, I think it's important to be able to understand how do you version it, how do you engage it, how do you improve it and watch it as you go. And it's hard to do that with everything, right? You really have to focus on certain products and stuff. So when I'm thinking about, and I think I will challenge at least the BICAM project in this space, is to sort of what are our next products and how do we make sure that people can engage them in effective ways. And if they're models, then making sure you have your MLOps and your access patterns correctly for that, and making sure you understand your drift or whatever it might be. And then there's another space where, like, there's a community of doing this work, and how do you allow them to and enable them to do that work as well? These are not consortia products, but these are very important space for people to explore that might feed into consortia products, might create models that everyone accepts and moves forward the ability for the consortia to move forward. So, like, both very important. Go ahead, Sarah.
Thank you. Yeah, just one other point, kind of slightly going off on a different tangent, but it just occurred to me, so I want to throw it in here, which is that for many of the cell compartments in the head, I'll put it, or in the brain, there are let's say cousins or brothers and sisters sort of in other tissues in the body. So that's immune cells, vessels, relationships. I mean, the skull, if you like, the meninges, and so on, and lymphatics, et cetera. And so I would like to throw out there that it is worth maintaining that plugging into the human cell atlas community would be my feeling for, achieving that cell annotation that's not just neuronal and glial focus. There's obviously the nervous system that's distributed across the whole body as well. But
right.
I just want to mention that while we're at it, kind of thing.
Yeah. And maybe Kimberly, if you might have some thoughts around that as well.
Yeah, so I was thinking something similar as well. I think that's one thing that comes to mind when thinking about integrating across the body is that there are commonalities across organs and tissues. And there's probably a need to think about those cell types within their different contexts. And so when we want to access the data, we might need to access data in terms of the tissue or region that it came from, but also if we want to think about physiology, there are other ways of hierarchically organizing the whole body, right, beyond just the brain. And so there also may be a need to organize cells within their cross-organ commonalities, essentially, is what I was thinking, which I think aligns with what Sarah was saying as well.
Yeah, that's exactly what I'm trying to say. Thank you for putting it so nicely.
I don't think I quite hit the mark, but maybe between the two of us.
Thank you so much. And so I'll just give a last moment, Jesse, if you're available, to talk a little bit about, if you'd like, about quality control when it comes to agents and AI and ML and hallucination and these kinds of things. I know, Sarah, you touched on several concepts around that. Jess, if you're interested in adding to that, you're welcome to.
Yeah, my answer is very predictable. Whoops, did I freeze? My answer is very predictable. Which is basically, I think touches on some of the themes that these are not new ideas, that we have the same problem with humans, which came up in the chat, that I don't really think that the answer is dramatically different. The problems we create are different, but the answer is always just independence, which has come up. What is the right class of test that is independence? And are we happy removing independence at each stage of our analysis? And so I just think that's a question we have to ask when we standardize the data to that middle bottleneck that Partha talks about. Are we happy that those standardizations are in fact universal and fixed and reasonable enough that we don't need to worry about them? Are we happy standardizing and using some universal training set for all LLMs going forward so that they aren't independent anymore. So, I mean, to me, it's just a question of at each step, you are choosing to lose independence. The word integration gets thrown around an enormous amount, but integration is not necessarily a good thing. You're basically killing independence in the data. So I'd be happier in a world that resembled a little bit the previous world, which is when two people who are, I don't trust any more than I trust an LLM, maybe less. I mean, humans can be held responsible, but humans have incentives too. So humans produce results, and I don't trust them at all. And I look at them and they're independent and they're competitive and I can compare and contrast. And I worry a bit that people will generate a universal data set. And listen, I'm super happy with the reference genome, but I'm super unhappy about reference bias. And it bedevils a lot of analyses that we then have to resolve subsequently, which again was a worthwhile trade-off. And I'm super happy with the reference mouse result, but I'm super unhappy that we end up with a lack of robustness to genetic perturbation of the class that we were interested in for translational efficacy. So there's just a question at each stage of an analysis, are we happy? Is it reasonable? And so that's my general answer to your question, Tim, which is boring enough that when I asked ChatGPT what I would answer this question, it correctly predicted this would be my answer.
No hallucinations today. All right. Thank you so much. So we'll jump ahead a little bit. Once again, this is all related, and we'll have time to come back to things at the end. And so for our third section, we're dancing around, even though we've talked about a little bit already, knowledge access and interaction patterns. And I believe I have Satra speaking a little bit about a slide. Is that correct?
That is correct.
Hi,
folks. Thanks, Tim. And it's very easy for me to talk about this slide, given all the panel discussions and the introductions to this overall section and themes. First off, I wanted to kind of point out that BICAN is in this kind of part of this broader ecosystem that the BRAIN Initiative has been planning and developing through various kind of transformative projects over the years. And within the BICAN knowledge ecosystems, we've touched on data repositories, brain atlas portals, knowledge bases, and a lot of the data pipelines and other things that have been talked about in this section kind of point to almost maybe whether it's somebody learning West Coast swing or building a plane as we are flying it. We live in a world where our standards, our process, and pipelines have to reflect some of the ongoing challenges in the ecosystem itself. And this relates to kind of knowledge access and interaction patterns. We touched on trust in a couple of the discussions in this last panel. Trust comes out of interacting with the system, understanding different components of it. Trust also comes out of the knowledge that we have to encode information in the system as data are generated, as knowledge is extracted, as pipelines are created. And so within this kind of thing, Jean talked about a need for an access pattern of a certain kind that's different from how a particular data repository might be thinking about access. There are pipelines that involve human in the loop, involve curation, that might require access and interaction with an ecosystem that's different from how we are currently doing it. Also, I wanted to bring up an idea. Tim has mentioned this word MLOps, and there's another kind of related idea in PsyOps that some of us have participated in putting out there is different levels of improvement in scientific processes in partnership with AI or other tools that are emerging online, and how to go from a process that was highly ad hoc to a process that's more automated and standardized in certain ways. And that's what an ecosystem really is figuring out how these different parts of the system fit together, evolve towards the benefits of downstream use cases that come from it. So with that, I'm going to perhaps pass this on to Carol to maybe discuss some of the questions for this panel, and then I think Tim is going to take over moderation around it.
Thank you, Satra.
Sure. So the questions that we have on this slide today are kind of these broad overview questions, and then our panelists are speaking to different aspects of these questions. And a lot of this has come up in the prior sessions about access and interaction patterns, what users need. So In this session, we're trying to cover what cell annotation knowledge might be extracted from brain cell atlas data, and the types of queries and searches that should be developed by data archives, cell atlas portals, and knowledge bases. And then really kind of summing it all up, we're trying to create this enduring community infrastructure. How is this developed that can allow brain cell atlases and knowledge to be advanced, versioned, and kept up to date? And so with that, I think we have some targeted questions to start working on this topic. Go ahead, Tim.
Thanks, Carol. And we've been talking a lot about cell annotation. It's critical and vital to what we're doing, and it's still part of our discovery. So maybe we can start there for a question for Evan. Since it was mentioned already, the awesome work that Evan and team is doing with CAP, which has been very exciting to see. In what way do today's brain cell annotations limit their use for future AI ML models where they scale to meet the needs of model development? Paola, thank you so much.
Oh, absolutely. First of all, this is such an interesting and stimulating discussion. And so probably what I'm about to say already has been said in some form or shape, and I just wanted to give perhaps my take. So as I think about what has been built so far, which is really exceptional in terms of data modalities, atlases, everything that we discussed so far, and where we want to go in terms of understanding biology and making predictions about how biological systems behave, and and be surprised by what we can really discover. I think a lot about perturbations, and so how perturbation will fit into these visions, because perturbations are really the place where we are testing and validating, what we discover and the role of things that we have discovered, to also produce knowledge that can feed and train the foundation models to make new predictions. And so one thing to consider, so multiple things to consider. The first is, what is the kind of knowledge that we need to find in our portals for that people can access about the perturbation. Perturbation data space is going to be so diverse and so messy compared to other data modality, right? That we really need to have a framework for nailing down the parameters of the perturbations, because the result of that perturbation will be dictated by the cell type you're perturbing, whether it is in vitro, in vivo, by the kind of manipulation you're doing, but who is doing it, at what time are you doing it, and so on and so forth. And yet, this massive data knowledge constitute an opportunity to really provide a lot of data to not only to models, also to biologists to think about, right, what this data really means. And the second thing to consider is how do we link, and I think Fabian made some comments yesterday about this, how do we link perturbation design to the kind of output data that the models require, right? So as of now, we can imagine a lot of different perturbations. Each and every one of us will think about them based on the questions we have, but that might not necessarily be the right type of data to produce for the community. And so I wonder if there is a programmatic upstream sort of gathering of thoughts about, can we at least control and the design of some of the perturbation to see if this idea that we can train foundation models to predict biological outcome will pin out, right? Otherwise, we're using whatever we have, and it might not be ideal at some point. And so I think that that's very important. And I'm going to stop there, but I think that thinking about perturbation it might be critical as we walk into the next stage.
Might be. Sarah.
Might be.
Yeah, I think it's a great point, that Paola's making. I just don't know whether in the brain, how easy is that? But for instance, in the human cell, as we're integrating all of peripheral blood data sets, and they're literally tens of thousands of samples now, and they cover a range of ages, sex, infection status, autoinflammatory status. And so for that, an admittedly much simpler system, you can And genetics I won't mention, but that's another dimension for where there's some data. Now you can do that in some sense, physiological state or perturbation to a cell ensemble, suspension cell transcriptomics, multiomics data, and that is very exciting actually. And begin to kind of observe human archetypes or groups of humans that have the same kind of cell ensemble status and demographic or environmental exposures and so on. For the brain, obviously there's a different scale of complexity, but I think we can start to think in that way and maybe start to move towards that. The most coarse-grained perturbation is obviously disease in human. In vivo, in vitro is easier, maybe.
Yeah, thank you. I want to share two very quick thoughts on the first 2 points on the slide, but really reiterating a lot of the exciting discussion that we already had, right? So I think the exciting time is, we're recognizing the comprehensiveness, we're enjoying the complexity of the system we're exploring, the knowledge we're accumulating, and then we're also running into this dilemma of the more we know, the less we know, right? So I think in the context of at this point, we really want to think about the balance. We want to find the right balance between the consensus or the reference. I really love the reference genome analogy. Without that reference genome, we would not have made all the exciting advancement that we made in genomics over the past 20, 25 years. But of course, the reference genome misses the variations, the dynamics, there are lots of things that it misses, but that doesn't take away any of the values of the reference genome. So I think it's about finding that balance. And we're building a reference, right? So we're building a reference, we're building a consensus, but we also allow the existence of variation and then the opportunity of exploring the variation, including perturbation. I really, really love that concept. In fact, that's also what's happening on the genetic side, right? So we're going from a reference genome to a pan-genome setting. We are taking advantage of that knowledge to interrogate functional impact of genetic variations. So the perturbation part of the human genome is happening. So I think that's a really good analogy here for thinking about what kind of annotation knowledge we want to extract. It's really about finding that balance, right? So by all means, we're building a reference, we're building a consensus, but we want to maintain the kind of the variation, the comprehensiveness of the system. So that's one point I want to make. A second point related to this is also something that I think got mentioned before. We are dealing with very different data, but the data really exist at different levels, right? So it goes all the way back to raw images behind the sequencing experiment, right? On one end, but the other end is the knowledge that we're gonna write into textbooks, right? So it really spans this entire spectrum. I think our job is to figure out the best way for people, users, investigators with different needs to be able to access data at different level, right? So I think that's the mindset, at least I have in mind, is to to have different type of accessing point to different level of data tailored towards different users and investigators. So that's what I want to quickly share.
Thank you so much, John.
Hi, yeah, that was great. Thanks. Getting back to Paola's point, Paola, I definitely agree that we want to be able to test these models with perturbations and in a bidirectional way. A couple things come to mind. One is, the importance of focusing on specific use cases to start. And one of the feedbacks we got from our multi-council working group was, as we develop these models, et cetera, to not do so in the abstract, to really think about specific use cases and tailor it. And I think that'd be a good way to validate. And also the importance of relatively, and I stress the word relatively simpler model systems. And I think one great example we have of how these kinds of data, this type of data can facilitate testing hypotheses and see how perturbations can also test the models is in the Flywire project, right? Where actually there's some really beautiful predictions that were made from the full connectome of the fly brain that could be tested with genetic mutations. And I think that's a beautiful example of how one might structure the data and the knowledge in a way that can be bidirectionally useful for both informing the perturbative experiments, but also have the perturbative experiments validate the model. So just a reminder to us all to think about specific use cases and the simpler models as we build this thing up stepwise.
Thanks for the focus, John. Dawen.
John, piggyback on your point of how the fly EM data, so the connectome data can be guided other further experimental design, and then come back to also these three questions that presented on the screen. That I feel that currently what limited us is that all the analysis pipeline or the ecosystem is kind of closed loop. So basically the lab or the consortium that created the data set that they would create the analysis pipeline as well. So it's a fairly closed loop analysis or software design. So we lack of something like ImageJ and Fiji. How is it so successful? It's because it's very open source. So everybody can write plugin for it. Everybody can develop and then to cross-reference and to test it out. So I think we probably need something like that from a concerted effort from NIH to help us to establish the ecosystem.
Absolutely. Carol.
Yeah, I think, while a lot of the BICAN infrastructure is this kind of closed loop thing, the moving into the knowledge-based world, where we're going, right, is where there's that opportunity to bring in multiple data types, multiple sources, annotations from different people. And I think in terms of creating this enduring thing, which we know is not one thing, it's a distributed ecosystem. There's multiple groups focusing on different things, expertise being applied to specific things. We've got really strong assets. We've got the reference cell types. We've got atlases. We have spatial resources. There's genetics, genes. Everybody talks about our genome references, which of course we know there have been multiple. The big challenge we have is growing this while it's changing, because we're at this point where the ability to integrate all this information is going to rapidly evolve our understanding of things. And so rigid metadata systems aren't going to keep up with the emerging knowledge. We've got many contributions. You have to sift through the really good stuff and then the less interpretable stuff. So it seems to me that in trying to make this enduring thing, we have to worry about how resource domains overlap. Different groups might create an authoritative reference on one thing. We want authoritative references to be recognized where there is that high trust, but while also allowing new things to come into that system. So it seems to me the major things we need to do are to support our user, to support interaction between systems, and to support interpretability, is having customs for provenance of where information's coming into the system at all sites that can be found across all the databases. So if you have an archive over here pulling in information from that knowledge base, you got to be able to track back where it came from. Two, you need to have some sort of designation on who is the current accepted authority. You can have multiple forms of authority that have slightly different flavors of information, but you need to start saying in your databases, who is your authority? If something's provisional, have a way to classify things as provisional, have ways to say, these are things that are not even provisional. These are so emerging, we're not quite sure what to do with it, but there's a lot of value in seeing it alongside your data. And I think the third is for our users is clear signposts on how you navigate the contents of these knowledge bases and other infrastructure pieces. How do you interpret it? What are you looking for to understand how usable it is and how trusted things are, and where's there that room to really still validate and interpret. So I think those would really help make this knowledge base infrastructure going forward to be something useful and dynamic.
That's well put, Carol. Paola.
Yeah, I couldn't agree more with what Carol just said. And I think all of these concepts and these principles are critical also as, again, we move towards knowledge base and understanding what things do, to perhaps leverage a lot of information that is already available in the field that, again, with the advent of AI agent that could perhaps facilitate at least a collection of the information, for example, from perturbation studies, for all sorts of things that are already available out there. And how do we integrate this knowledge into our systems by also giving them a grade, right? Like you said, Carol, is this stuff that is just emerging? Is it stuff that we haven't been able to double check, but it might still have a value, especially in the context of a lot of data to train models. And then the last point I wanted to make is about prediction of one modality outcome from studying a different modality outcome, which might be also a way to think about how do we connect the different type of modalities of data that we have. And there is not a single, opinion about how to do that, but this is a major goal in biology right now, that people are trying to predict what a functional circuit level outcome might be from analyzing large amount of, let's say, molecular data or whatever else. And so thinking about also, this is more sort of in the longer term, very long-term future, but this is what we're thinking about right now as we think about the evolution of the infrastructure. Will we ever can we be in a position where we can try to see if we can predict one modality from another? Because if we could do that, then perhaps we don't have to collect as much data, just as a thought out there.
Very interesting. I know we wanted to potentially have group conversations, or at least the ability for others to ask questions. So we're running low on time for that. So let's quickly make sure Partha and Maryann can go through and ask their question. Then maybe we will have a time for at least one question from others. Partha.
I'll just quickly say, going back to the human versus automation loop question, Richard Sutton has this thing, he calls it the bitter lesson, which is essentially get the human out. And what I want to say is that that's for engineering. I think in science that doesn't apply, and we need to keep the human in, but we do kind of can use these black box techniques. Where the human comes in, I think, is the larger, bigger picture architecture of, okay, I have this black box that does something specific. It maps between atlases, whatever. And I don't need to look inside that black box, but I may need to put a trust layer around it. But where the human really comes in is what is the goal of the overall system, and what is the bigger picture architecture, the system architecture of the whole thing. And that's where I think the human is really necessary.
Yeah, in an agentic world, the scientist becomes the conductor and doesn't play the instruments, right? Maryann.
Yeah, just wanted to make a brief comment on to follow up on what Carol and others have said. I think the FAIR data principles, which really emphasizes these identifiers tied to rich metadata, do provide the capacity to add what I call these little stable nodes, right, inside of a very dynamic ecosystem. Owen, going back to what Owen said, right, the archives are immutable, and those data sets, those raw data sets, get deposited, and they are a point at which then the community uses those artifacts to produce other things. But what I've noticed over the years in working in this, and I think that this is something that will have to change, that the way that researchers deal with data, it's somewhere between a research product that's now going to need to be shared, but also an asset that they use. And therefore, when they do share data, it is not treated in the same way that you would treat a scientific article, where it was really, designed to communicate to others and wrap it up. I do think that's going to need to change in the age of AI. and that we will have to learn about things just like the publishing industry did. What is a version of record, right? Where did this enter the ecosystem? And then make sure that we reference back to that, just like we have a system of referencing articles and other things to say this is where it came in. That allows both these midpoints and other points that we can verify, look at, say what did this start at, and also provides then for more automated means to start to track how provenance is how these things evolve over time. So I do think that that is something we have to think about. We're making progress on it, but it is still a little fuzzy, I think.
Yeah, I'm looking forward to the community thinking more deeply about fair agents, fair models, fair knowledge, moving those concepts. All right. Well, everyone has the opportunity to continue this conversation in the question and answer. And I would encourage our panelists to keep an eye on that and please respond and engage questions as they come up. So just a big thank you to our panelists and a big thank you for the opportunity to be able to give space for this conversation. It's super important, and I think we maybe scratched the surface, but I think we at least named some of the things that are important to carry on in the conversation as we move forward. So thank you everyone for your time.